Well, I was asked to talk about scales and orders of magnitude. And when Dan asked me to talk on this subject, he said, can you talk about scales? So I said, sure, I'll be glad to talk about scales. And I'm having the same problem here. So there's a scale, but I don't think he wanted to talk about that one. There's another kind of scale. This is getting close to the kind of scale that we might be talking about, but in fact, I'm going to be talking about scales with distance. Oops. So here is a picture of Camp Randall. Okay. It's not a very good one, and it doesn't, it's a bad scale, isn't it? So maybe we can zoom in a little bit. And the first one was taken from 20,000 miles away. This one is, I guess, 2,000 miles away approximately. Go in another factor of 10, we're getting a little closer, another factor of 10, and now we're down there. So what we've been talking about here is making measurements and relating them to each other by a factor of 10. And so we talk about that as an order of magnitude in the jargon. Now, I'm a biologist, so what I'm going to be talking about is things that relate to biology. We all know orders of magnitude, even just in a meter stick. A meter stick is a meter long, but 100 centimeters. This is 1/100th of the stick. That's a tenth of that, and so forth, and you can go smaller and smaller. And in the world that we live in, we normally deal with things that are in the range of a meter. We're 1 or 2 meters tall. An apple is not a tenth of a meter, it's a little less, but it's at that order of magnitude. A wasp is a tenth that size, and so forth. And so we can relate to things that are orders of magnitude even in our own existence. Up here we have a scale that's relating to meters. 1 meter, a tenth of a meter, 100th of a meter, 1,000th. And so the exponent is just the number of zeros after the decimal point. So we can see these things with from the human down to even the hair, if you have it. We can see these things with our naked eye. But if we want to see any more structure, the resolution is not great enough. And so we all know what happens. What we want to do is look at some of the other stuff that's smaller. But even though it's small, it's pretty important because these guys can kill you. So I'd say it's important. So we want to look at these things, and we use other ways of doing that other than just our naked eye. For example, the magnifying glass is a pretty good one, and that goes fairly well. But better than a magnifying glass is the microscope, which is like pretty much a fancy magnifying glass. A microscope has lenses in it, just the way a magnifying glass has. It has a source of light that shines through a sample, and the light goes up through magnifying glasses up to where you look in. And so what you look at, if your eyeball is here, you're actually looking at the actual sample that was down here on the stage. And so this helped enormously in our understanding of small creatures that are around us and in us. For example, here is just a sample of some cells. This is about 10 times 10 to the minus 6 meters. That we call that a micron. So that would be 10 microns in this bar here, and that's about the size of the cell. The nucleus is the red thing in the middle. That's where all the DNA is and the genetic material. And there's cytoplasm on the outside of the cell. Okay, well, that's good. You can see some resolution there in this cell looking through the microscope. But if you wanted to have more resolution, you could see more into the cell. You can't just project it and make it bigger because all you do is make it fuzzier. And the reason you make it fuzzier is that what we're really looking at is light. And it's light that's coming through, visible light, that's coming through your sample. And so this is the spectrum of the visible light. You see that with the prism. And that's just part of the electromagnetic frequency that we have. And this is the part that goes through the microscope. And if you want to resolve something, it can't be significantly smaller than the wavelength of the light that you're using to measure it. And so if we wanted to measure something that's much smaller than the wavelength of light, we just don't get that kind of resolution. So we have to use something else that has shorter wavelengths and therefore better resolution. And one way of doing it is X-rays, and another one is using electrons, which also have a frequency, and we can use the electrons to probe some sample. And in doing that, we use what's called an electron microscope. And so here the device shines a stream of electrons down. The sample is down here at the bottom. And so these electrons are focused by electric currents in the tube. This is the actual device here. And it is shined down to the sample, and then the electrons are collected, and we can look at the image. You're not actually looking at the sample now. You're looking at an image of the electrons that were able to go through the sample or reflected off the sample, and that's picked up electronically. What this does is it gives a huge increase in the power of resolution for looking at biological samples. And for example, here we have the head of a mosquito. Okay? This is an electron microscope. All these little dots over here on the right are very primitive eyes, and each little dot gives a little signal of it's the way that the mosquito can tell distance or at least detect light, and with all those eyes, no wonder you get away from you. Okay, well this is a fun picture to look at, scanning electron micrograph of a mosquito head. This is about the kind of resolution you could get with a light microscope, actually. It was not, the idea was not to push the resolution with this picture. But now if we look at just in a localized region here and amplify it, magnify it, we can see that this little region here, when we magnify it fivefold, now we can see all sorts of other structures in here. And so this is a way that the electron microscope is able to give us a lot more resolution about and understanding what biological samples really are. Well, that's fun looking at a mosquito. Here's a cell, and with the electron microscope, we can see details of the cell that we couldn't see with the light microscope. This is not the cell. This is the nucleus of the cell. The whole cell here goes all the way up, all the way around, fills the whole screen here, basically. And now you notice that this bar here, the blue bar, is about 10 microns, which is the same scale that we had in the previous picture. Okay, so it's 10 microns, 10 to the minus 6 meters. This is a picture of E. coli bacteria. These bacteria are much smaller than the cells. These are individual cells that you're looking at here. each one of those little sausages is a cell, a bacterial cell, and they aren't actually this color. That's just the way you feel if they're in the wrong place. But they're about maybe 2 microns long, 2 to 4 microns long, and half a micron in diameter. Much, much smaller than the cells of the higher organism like us. So cells have different sizes depending on the organism we're looking at. Viruses are even smaller than bacteria. And these are just a couple of viruses that papillomavirus and herpes viruses are human viruses. And again, the scale is much smaller now. And so we can see that these herpes viruses are 100, what we call 100 nanometers. That's a tenth of a micron. And the papillomavirus is even smaller. And these are electron micrographs of the viruses. Now, with this kind of magnification, now we can start to see some structure in the viruses, see what they're made of. You can see all these little, probably hexagons in this. These are individual protein molecules, and likewise in the herpes virus, you can see individual protein molecules, and you can also see some structure in the way the protein molecules are assembled. Over here on the surface of the virus. Well, can we go even further? Can we ask questions about even more detail of these biological entities? And the answer is yes. And now we can use yet a different kind of technique, which is Xray diffraction. And in Xray diffraction, there's a source of X-rays. Nowadays, people go to accelerators like the one outside of Chicago, put a biological sample in the accelerator, a crystal of that sample, and that sample will scatter when the X-ray beam hits the sample. The X-rays are scattered by hitting atoms in the sample. And these scattered X-rays can be captured by a piece of film, X-ray film. Nowadays they do it electronically, but it can also be used just with a piece of film, that each one of these spots represents a reflection of some atom, of an X-ray that hit an atom in the crystal. And now to get from that kind of structure to that kind of data, this is data that one gets. There are ways that you can calculate the structure of what's in here just from the way those X-rays were scattered. Okay, I won't go into that, mainly because I can't do it myself. Down below here, this is the X-ray of probably a protein crystal, I'm not quite sure what. Down below here is the diffraction pattern of DNA. And this was actually the pattern that was used in elucidating the structure of DNA about 60 years ago. There's a big difference in the resolution, obviously. One of the example of what you can learn from this kind of analysis is, here's a, whoops, this darn thing is, this is a particle that's in all of our cells. It's called a ribosome. It is a particle that is responsible for making proteins. And this very complicated machine, it's got RNA and proteins, and you can see individual atoms now in this very complicated structure. And now again, the scale is blown up so that we can see the whole molecule. Finally, I just mentioned DNA. So we have the structure of DNA here. It was elucidated 60 years ago from yesterday. is my understanding. DNA itself is a very long molecule. You heard this from Lloyd Smith at the last one of these sessions. It's only about 2 nanometers across in diameter, and this is the size of each base pair in the molecule. There are enough base pairs in the DNA molecule that we end up with about 2 meters of DNA in each one of our cells. Okay. So it's a very thin molecule. That's how we can squeeze all that DNA into the nucleus of our cells. Just out of a curiosity, if you do a calculation, you can, since we have about 10 to the 13th molecules, 10 to the 13th cells in our body, take the 10 to the 13th times the 2 meters that are in each one of our cells, and that's how much DNA we have in each one of us, okay? And if you go through the calculation, that's enough to make 65 trips to the sun and come back, okay? Which leads me to the beginning of my talk, which is we're all related in some way, in that way sense, and I think that leads me to the next talk, which is on looking at big distances rather than small distances. Thank you.